Students, welcome you all on the behalf of Rahul Anand Tutorial. So as you know that I am Rahul Anand and this is somewhat around my channel whose name is actually the Rahul Anand Tutorial. So now let's begin with a very interesting topic which are often being asked in terms of your Neet, often the Aiims, often the All India Pre-Medical Examination. So these are literally belonging from which of the chapters of your book? So these are belonging from the molecular basis of inheritance. So now today we are going to deal with the very interesting topic. So what are actually these? So these are the Hershey and Chase experiment. So primarily, when I am talking about the Hershey and Chase experiment, the thing you have to remember that what? So Hershey and Chase performed an experiment with what? With actually the bacteriophage or either the viruses. Remember? So the bacteriophage is equal to viruses, which is having the potential to multiply as well as grow within what? Within actually our host cell or either the bacterial cell. Fine. So while this experiment was being done, so it was being done to prove what? To prove that what? That actually our DNA is the genetic material. Fine. Now, if I am talking about the DNA, I mean the genetic material, so it is composed of what? So it is composed of the two polynucleotide chain, which is often known as the ribbon-like structure. Fine. Which is having what? Which is having the nitrogenous base. And in the case of here, I mean the DNA, you will be having what kind of the nitrogenous base? What? So it will be the adenine, guanine, as well as what? The cytosine as well as the thymine. Now, so these are being attached with one another by the help of what? I mean our adenine will be attached with the thymine with reference to the double bond, as well as what? The guanine as well as the cytosine will be attached with one another by the help of the triple bond. So these are actually what? So these are the nitrogenous base of what? Of actually our DNA. So now let's proceed with reference to these here in the Hershey and Chase experiment. So if I am telling you about the Hershey and Chase, so these are not actually one name of the scientist. So these are what? So these are actually the two broad type of scientists having the huge bulky quality of brain. Now, so let's try to discover. So Hershey plus Chase means what? These are actually two scientists. Now, so they use two things for what? To find that DNA is the genetic material. So what are actually these two things? Can you tell me? So these are the E. coli as well as what? As well as the T2 phase. Now, so if I am telling you about the E. coli, so what are actually these? So let's try to elaborate with reference to these names. So E means what? It is the Eusteria. And what is actually these? So these are the genus. Then what? What? Then the C. C means the coli. Fine. And here C means the coli. So these are what? So these are actually the species. Then what? Eco, right, means the R. So R is here representing what? So it is representing the what? The strain, which will be actually rough in structure. So you could often probably find here on the board here. So you could find that it is having the strain which are quite rough in structure. Now, then what? Then the I. So here I is the Roman letter which is playing an important role to describe what? Which is playing an important role to find that in what era actually the enzymes were isolated. Find here. Describe the order in which the enzymes were actually isolated. Here. Now. So when we are talking about the T2 phase, so T2 phase will be directly proportional to what? It will be directly proportional to the bacteriophage. As well as bacteriophage are directly proportional to what? So it is directly proportional to the viruses. So when I am talking about the bacteriophage, remember what are actually these? So here, the bacteriophage. So viruses that multiply inside the host cell or under the bacteria cell coming under what? It will be coming under the bacteriophage. Now, so let's try to elaborate what are actually the names with reference to these in the bacteriophage. Bacterio means what? The bacteria. As well as the phage means what? To eat. Here. Now, so let's try to understand what are actually these. In terms, you will be able to understand logically what are actually the resemblance to the in terms of what? The structure. So it is having what? It is having the body structure. Likewise, the tadpole. Find here. The tadpole shaped structure. Now, so when we are talking about the head, so it will be actually what? So it will be actually the hexagonal in structure. Fine. With reference when we are talking about its tail, so it will be what? So it will be actually what? Cylindrical tail in structure here. So now, here. Now, so let's try to discover what are the different steps which are being induced, as well as what are the different cases coming under what? Coming under actually our Hershey and Chase experiment. So these are what? So these are the what? These are the here mean the absorption. Then what? Then the injection. Then what? Then the replication. Then in final you could find here mean the lysis process here. The lysis. Now, so when we are talking about these, means what? Actually the first cases, mean the case first. So these are what? So these are actually the adsorption. What will occur here? So in this case the bacteriophage as well as what? As well as the wire cells will attach with the host cells or rather the bacteriocells. You will find here. Here. Now, so when we are talking about the second step here, mean the injection. So in this case what will be happening? So in the case of the injection, the mean the bacteriophage will start to attach as well as penetrate what? Penetrates their DNA into the host cell. So when we are talking about this, mean the injection. So what will actually occur in these cases? What will occur? So in this case the bacteriophage or rather the wire cells will start to penetrate its what? Deoxyribonucleic acid into the what? Into the host cell to the third stage. Means the multiplication. So fine. In the case of this, mean the replication, what will occur? The bacteriophage will start to multiply. Where? It will start to multiply as well as replicate into the bacteria cell or rather the host cell. Why is it so? This is due to because you know about the DNA. Fine. So you could go before in terms of my lecture tutorial. I have dealt with this topic. So there I have made you explain what are actually the DNA, mean the deoxyribonucleic acid. So these are composed of the polynucleotide chain, often having the nitrogenous base. What are actually these? These are the adenine, guanine, cytosine, as well as the what? The thymine. No. So due to because the DNA is the genetic material and it is present inside what? So it is present inside the bacteriophage or either the host cell. Thus what will happen? It will start to multiply down due to the sufficient amount of accommodation of the life supporting mechanism. And often you know that DNA is actually the genetic material. Means our bacteriophage or rather the wire cells will start to multiply as well as replicate. Where? It will start to multiply into the host cell or rather the what? The bacteria cell. Now, so let's try to proceed with the fourth thing here. Mean the lysis. So the by the name it is very clear that something which will start to break down. Why is it so? Let's try to get the concept with reference to the another example. So we are having the electrolysis. Means what? Something will be breaking down in terms of what? In terms of our electricity. You are often having the hydrolysis. Means what? Something will be breaking down in terms of the presence of water. Then what? Then you are often having the meiosis as well as the mitosis. Meiosis means what? Meiosis means what? The reductional division generally takes place in terms of our gametophytic stage or rather the gametophytic phase. Another meiosis means what? So meiosis means the here means the reductional division. Then mitosis is what? What? So it will be the equatorial division. Fine. So likewise you could understand that lysis. Okay. So it means what? To something break down. Fine. So in this case, what will happen? Due to the multiplication of what? Due to the multiplication of the bacteriophage or rather the wire cells inside the bacteria cell or rather host bacteria cell or rather the host cell. What will happen? There is a bulky amount of what? The bacteriophage or rather the wire cells will be present inside the here, mean the host cell or rather the bacteria cell, and it will start to exert the bulky amount of pressure or rather the force into what? Into the wall of the bacteriophage or rather the host cell. Thus there will be the phenomena like such as the lysis means what? To break down will occur. So here the breakdown of what will occur? So here actually our mean the bacteriophage, bacteria cell or rather the host cell will be breaking down. Due to what? Due to the amount of the force exerted by bacteriophage or rather the wire cell. So you could popularly find here on the board here. Now, so thus if the wall will be breaking down means what? The wall of the bacteria cell or rather the host cell will be breaking down. What will happen? The bacteria phase as well as the wire cells will come out. Fine. So this is known as the case of the lysis. And something I have left. So I will be making you understand in my next lecture. Otherwise, you will not be able to get that. So fine. So I thought you have got the concept. So what are actually these? Mean the amount of Hershey chain experiments, as well as they use what things to proceed with these experiments, such as what, such as the T2 phase, the cryophase, or other the wild phase with reference to the ecodye. And ecodye are what? So you have understood now. Fine. So I thought you have got the concept. What actually these? Mean the bit amount of introduction with reference to your Hershey chain experiment. So, if you like the work with reference to the education, often the lecture, so you could often subscribe, like, as well as comment below in the text. Thus, I will be knowing that what concept actually you are hungry for, or whatever you understood or rather got. So, very thank you with reference to what? With reference to the Rahul Anand tutorial. Very thank you.